and welcome to Let's Meet the Virologists, a podcast about the people behind today's virology headlines, people working to understand viruses and how they affect you. We are talking with students, postdocs, and other virologists so that you can learn who they are and what they do. I am Larissa Thackeray, and I am hosting this podcast from America's Heartland in St. Louis, Missouri. On September 9th, 2022, we talked with Dillon McBride, a fellow at the U.S. Department of Agriculture's National Bio and Agro Defense Facility Scientist Training Program, and a PhD candidate in the Department of Veterinary Preventive Medicine, The Ohio State University, and Dr. Leonardo Caserta, a postdoctoral associate in the Department of Population Medicine and Diagnostic Science at Cornell University, about their work on white-tailed deer as a reservoir for emerging and historical strains of SARS-CoV-2.
Thanks for joining us today. Why don't you guys tell us a little bit about yourselves?
Yeah, sure. So my name is Leonardo Caserta. I'm originally from Brazil, and I did my graduation in veterinary medicine in the State University of São Paulo. And then my master's and my PhD I did in the University of Campinas, also in the state of São Paulo, and always focused on virology. Initially, I was working mostly with avian viruses, and then I started working with wild animal viruses. And then while I was finishing my PhD, I came to the United States to do my postdoc in Dr. Diel's lab, initially in South Dakota, at South Dakota State University. And then we moved to Cornell University, where now I work with mostly with sequencing of livestock viruses, but also wild animal viruses, and also development of recombinant vaccines.
Cool. And before we get into some of the science that we're going to talk about today, Dillon, can you tell us a little bit about yourself?
So my name is Dillon McBride, and it's super nice to meet you, Leonardo. I'm a PhD candidate at the Ohio State University. I started my bachelor's at Ohio State, mostly interested in organismal biology, ecology and evolution, and I sort of stumbled my way into some undergraduate research that's more on just diseases, and I really found a lot of passion in disease ecology and the evolution of infectious diseases. And so when I finished up, I started my PhD there with Dr. Andy Bowmen, and we really are kind of human-animal interface specialists. So we're nested in the Department of Veterinary Preventive Medicine, and we focus on zoonotic diseases, and we do a good job really sort of narrowing in on where that interface is. So pre-pandemic and ongoing for my doctoral work, I focus a lot on influenza virus that transmits from pigs to people. And of course, since the pandemic, we've been embroiled in the SARS-CoV-2 work in white-tailed deer, a little bit of mink as well now, but that's sort of where we find ourselves in the greater picture of that public health scope.
Cool. And I guess just to sort of let our listeners know, why did people start looking at white-tailed deer in the first place? So I know, in the beginning, people were kind of interested in, the origins of SARS-CoV-2 and, what animals other than humans were infected, but why white-tailed deer?
It started with computer modeling of the ACE2, right? So very quickly, researchers identified that the ACE2 receptor seems to be what SARS-CoV-2 binds to. It happens to be pretty conserved across mammals. There was a lot of in silico work. I know people point to the Damas et al. Paper kind of as the beginning. They basically, it's a little bit limited. They used a small binding region of the ACE2 receptor and identified some mammalian species that they thought might be susceptible. And then ongoing work with susceptibility. Leonardo is a huge reason, right? They did some experimental work where they really showed how efficiently they could be infected experimentally. There was also some USDA work along those lines. And then the Richt laboratory at K-State was also managed to do some experimental work along the way. And so we had a project that was funded internally, basically just doing both environmental surveillance and animal surveillance. And the idea of this virus seems to be a generalist, right? All of this precursor work is really showing that. And so let's take a look and see what kind of animals we can find it in: domestic animals, zoo animals. It's also the project that sort of started our wastewater surveillance here in Columbus. And it included white-tailed deer. largely in part due to the modeling of the ACE2 and the experimental work that really showed that there's a low transmission barrier there, right? They got infected, they shed it, they can spread deer to deer. And yeah, we had a lot of partners all over, lots of different divisions and other faculty members in our department that were helping us collect samples, and we were getting lots of them in. And funny enough, when we finally found it, it was our last batch of samples. We were ready to tie a bow on the project and wrap it up, and we got our last 360 samples, and 129 of them were infected with SARS-CoV-2, and here we are.
Great. And Leonardo, what do you have to add for that? Why deer?
Yeah, there is also the fact that there are 30. The estimated white-tailed deer population in North America is 30 million animals. So it's a mammal that has a large population, and because of hunting, there are a lot of chances of contact between deer and humans. So it's always interesting to look for viruses in these kind of animals that are always in contact with humans. And it's a very adaptive animals you can find in urban areas, in crops, in the middle of the forest. So these are animals that have large chances of acquiring human viruses.
Right. So Leonardo, can you tell us some of your early work with the deer? What did you guys find?
Yeah, so our group did experiments with deer, infecting intranasal deer with SARS-CoV-2, and we found that they will shed virus during a short window of around 5 days, but they don't show clinical signs. It doesn't mean that they don't get sick. It doesn't mean that they're not having symptoms, but for animals we say like clinical signs, right? So it's difficult to see if an animal is actually sick. But then, yeah, we found that deer are very susceptible to SARS-CoV-2.
And this was through an intranasal inoculation, or how are you giving it to the deer?
Yes.
Is that the way that they're thought to naturally get infected intranasally, or is it known?
It's not very clear, but they can get infected by baiting, because we know that hunters bait deer, so they can get in contact orally with SARS-CoV-2, but they can get in contact with themselves by touching noses, by being physically close. So it's also possible they are also get infected by nasal contact.
And do you know from your studies what tissues are actually infected in the deer? So where do you find the virus?
Yeah, there is a large tropism of SARS-CoV-2 in deer, but are mostly in the respiratory tissues, but also nervous system. Virus was found in nervous system, but SARS-CoV-2 can be found in a large number of organs in white-tailed deer.
And then Dylan, can you talk a little bit about some of your work sort of showing how the virus was getting into the deer, and then how the virus is actually, I guess, evolving in the deer, and what that might mean for sort of the chance of a new spillover from deer to humans?
Sure. Yeah. I mean, first of all, I want to echo what Leonardo said about, deer as a reservoir species, right? What he said about distribution and abundance, and as a generalist species too, especially, right? Like he said, using all sorts of environments. I mean, it's such a good point when we're talking about wildlife reservoir establishment. And, tissue tropism too, right? He said all kinds of tissues, and right, namely lymph nodes, which has allowed people to use, like in his most recent preprint, chronic wasting disease samples, because they collect referopharyngeal lymph nodes for those samples. And so really allowing us to use the strength of funding, programs that are already established to be able to get samples has been super critical for characterizing this system. And yeah, it's a great point about exposure route. I think it's everybody's $1 million question at this point. Leonardo mentioned baiting, interestingly enough, it's not legal to hunt over bait in all states, right? So there are states where hunters aren't baiting deer. Likewise, we have our upcoming year of surveillance, right? Our first initial discovery was really just in this like metropolitan park area, right? So we had 9 reservations from within one park system. And then after that, everything sort of spiraled into a much bigger effort. And so much like Leonardo's preprint, and which I'm sure he'll talk about, we really went out to try to cover the state, right? Try to find as many counties as we can. We're collecting hunter killed deer, hunter harvested, agency culled, right? So anywhere where hunting is not permitted, we have agencies that do population control. That first paper that you saw that caused you to reach out to Andy, right? That was all agency culled deer, but now we have hunted deer as well. And so they're not all baited. People say wastewater, right? Water runoff is a crowd favorite hypothesis, but nobody has really been able to isolate SARS-CoV-2 in wastewater. So that poses an issue. We've heard intermediate species. People think maybe rodents or bats, but then you have to explain an interface of rodents and deer, and then rodents and people as well, which is not exactly an easier causal pathway to explain. Contaminated trash, contaminated bait is a possibility. Fact of the matter is we actually don't quite know. So our most recent year of data, we collected 1,500 nasal swabs from 83 of Ohio's 88 counties. And I think we detected 32 independent introductions based on sequence, right? And so it's only in like 3 months, and we have 32 introductions from people to deer based on the sequences we can see. And so not only is this exposure route apparently happening, right? It exists. The barrier is low, but it also seems to be everywhere, right? It's all over the state. I think USDA has it in 26 states now as well, right? 26 states across the United States. And so it's everywhere. It's all over the country. It's in urban centers and rural areas. And nobody really has any super solid ideas of how deer are actually getting it from people.
Right. Is it viremic in deer? So when we always think with like flaviviruses, we always think about ticks and deer and things like that. Is it possible that it could be transmitted by ticks?
That's a good question. I'm not sure if anybody is really checked. I'm personally not super well versed in arboviruses, but I know that there's a little bit of distinguishment between infected blood in a tick versus a tick that is actually infected, right? So some pathogens have a life cycle in the tick. I don't know. We haven't tested blood samples virologically. We did collect some for USDA's larger nationwide project. I think they're doing antibody surveillance, right? They're looking at seroprevalence. But we're collecting them on filter strips, right? So we're for sure desiccating any viral RNA that might be there.
What do you think, Leonardo? What's your favorite, or what's your favorite hypothesis?
Yeah. So my initial thought was also wastewater, but then it started to become clear that wastewater is not so easy to find infectious virus in wastewater, right, like Dillon said. So the most probable route is human, not indirect contact between human and deer. But it's not clear which one. But I think it's something that soon will become clear. I think it's a recent topic, so it is something that requires more study.
Right.
And we're having a better idea anytime soon.
Right. And your recent preprint, I was very interested about how you were finding sort of the deer are not just a reservoir of virus, but they're a reservoir of almost like extinct viruses. Can you describe that work a little bit?
Yeah, it's very interesting. So we also used samples that were collected for chronic wasting disease surveillance, more than 2,000 samples in the hunting season of 2020, and then also more than 2,000 samples from the hunting season of 2021. And basically the virus was found all over the state, in 57 out of 62 counties in the state of New York. And it was very interesting to find, especially for the gamma variant. Gamma had a very low prevalence in the United States and also in New York State, but we found a cluster of gamma variant in one county, Allegheny County, in the south part of the state. And it's pretty interesting to find that, because it's a virus that it's that is not circulating anymore in humans, or maybe very extremely sporadic findings in humans, and also alpha variant and delta variant. Delta was still circulating recently, so it's not a huge surprise that we would find this virus in deer, knowing that deer can be infected by SARS-CoV-2. But alpha, there were, I think, very few sporadic detections in humans in October last year and August. But the peak of detection of alpha in humans was during, I think, between April and June last year.
Yeah.
But this variant was found very spread across the state. So it's so deers are not only reservoir for SARS-CoV-2, but we show that they are reservoir for nearly extinct variants.
So how do you think that's happening? Is it just that there's not almost like the evolutionary pressure in the deer? I guess you could sort of argue in a way that in humans, the new variants are chasing out the old variants, as it were, or just everyone's getting infected. And so basically there's immunity to that variant. So what does it mean that deer are actually harboring these sort of older, more ancestral strains, or continuing to harbor them? What does that mean?
Yeah, so probably deers, as soon as the pandemic started, as soon as there was a broad circulation of this virus in humans, deer started to get infected by SARS-CoV-2, and then because of the large deer population, there was a transmission from deer to deer, and these variants became established. So of course there was some containment measures in humans, but it's totally impossible in deer. So they just keep doing their natural behavior. So all of these initial variants, not only alpha and gamma, but those initial variants, the B1-like variants, they are still present in deer. So because of the large population and the impossibility of virus containment in these animals, because of the natural behavior, these variants keep circulating in deer.
I see. And Dillon, so your work kind of showed, though, that there definitely is evolution of the virus. So you're getting amino acid substitutions in the viruses that are circulating in the deer. So what does that mean for sort of potential spillover or back spillover or this kind of a thing?
Sure. So it's a great point, and I really like what Leonardo said about persistence, right? It's very difficult to control deer to deer transmission in a wild population, especially with so many deer. And I think it really lends to the point of, we really are right at the beginning of this, right? We're still seeing all these fresh spillovers, but we haven't really had the longitudinal time yet to really see if one of these is going to be the one that establishes in deer. And you start to see that like 5, 6, 7, 8, 10-year, decade-long persistence of a true deer origin lineage, right? So we don't have multiple years of evolution in a deer yet. We still have all of these introductions, and just why, we're seeing Delta, we're seeing Omicron, like this flavor of the month style spillover, but we're also seeing some of these select older ones, and they are evolving, right? I mean, branch lengths are long, right? When you look at the phylogeny, the branches are really long. They're very clearly distinct from the human precursor viruses. We have not identified any potential zoonotic events, any spill back into humans yet in our sequences. There was a preprint out of Canada that had sequence homology that potentially pointed to maybe one human case, a human sequence that existed at the end of an extremely long deer branch, which is the first limited evidence of possibly a spill back into humans, but no real evidence of onward human to human transmission. We do have some work that we're working very hard to get compiled and to get out soon. I think we all know the struggles of trying to publish collaborative research. We have a cross-institutional collaboration where we did take some of our deer viruses and isolated them and took a look at true spillback risk, some in vitro and in vivo work, serum neutralization from hamsters vaccinated with human precursor Delta, or sorry, not vaccinated, hamsters inoculated with human precursor Delta, human precursor Alpha, as well as like the Pfizer strain mRNA vaccine, right? And so testing serum neutralization from those different, right, anti-seras against our deer origin alphas, our deer origin deltas, and seeing how that does. And then as well as in vivo work, right? Hamsters that are truly just vaccinated with the Pfizer vaccine and then challenging them with a deer origin virus. And with a couple of exceptions, right? And we're still working on getting it together. It does, for the most part, look like there's not, like there in many cases, the replication kinetics are actually worse in human cell lines compared to the human precursor viruses. There's not a huge risk. There's not any of our deer viruses that seem to be super immune escapee or anything like that just yet. But just yet, I think is the key there. The branch lengths are long, which means these viruses are changing with time. They're evolving independently and in parallel to a human outbreak, right? So you have this backdoor evolutionary pathway for new changes. And even though it doesn't seem to be super risky right now, I think the real risk is establishment. It seems to be a COVID in 10 years problem, less than a COVID in 10 months problem. And we're working really hard to try to address that. We did see some select amino acid changes. It seems to be evolving, but not under super aggressive selection pressure. And I think that the number of spillover events that we see in all of these variants spilling back combined, like when you look at the evidence holistically, it really points to the interspecies transmission barrier must be so low because it's happening so much. We're not seeing any really specific unique mutations that are precursors to spill over into deer, nor any really aggressive selection pressure once it's in deer and transmitting deer to deer. So it seems like that barrier is just so close, which could point to it being risky in the future if spillback is also just as easy with such a low barrier.
Right. And trying to think, has there been in deer at least sort of a parallel of another virus or pathogen that was sort of spreading, and how long did it take to basically spread, throughout the deer population in North America? Is there any sort of modern parallel to this?
There's epizootic hemorrhagic disease virus and blue tongue virus that cause like really high mortality in white-tailed deer, but it's so high mortality that I don't know. We're talking about a virus that for the most part infects subclinically, like Leonardo said. So I don't know how parallel it is. USDA has been fighting the brucellosis and bovine tuberculosis fight for decades now, and white-tailed deer still pose as a challenging reservoir for that, right? There's still sporadic spillover into domestic animals. There are still cattle outbreaks linked to white-tailed deer in those little pockets of TB and tuberculosis and cervids in North America. So I don't know about how long it would take to rip through a North American deer population or what the reinfection risk is going to look like. I don't know if Leonardo has experimental work upcoming on deer reinfection. I know these deer transmission studies are hard.
Yeah, that was actually my question. Do you know, is there any immunity or can the deer be reinfected again and again?
Yeah, I think this is something to be tested. This is probably something that we're going to look at, but it's also not so clear right now. Okay. But it's definitely something that we must look into.
Yeah, and I guess sort of following up on that is, why do you think, as Dillon was saying, the infection is relatively asymptomatic? So, in humans, obviously, there's a huge range of disease. So people that are truly asymptomatic to people that are, dying. So there's an enormous range. Why are we not seeing that in deer?
Yeah, that's a good question. I'm not. I've afraid to say that I'm not so sure.
Does the virus not replicate to high titer in the deer, or is there some other deer immunity? I mean, I don't know.
I almost have a hunch that it's just it could, I certainly don't know from a biological standpoint, but at least from a statistical standpoint, it might just be a sample size problem, right? I mean, we see people all the time. We live with people. We see them get sick. We see them get hospitalized. I know that this experimental deer work is hard. I know that it is expensive. Connor Cool, who was with the K-State group, talked about challenges posed by doing, respiratory disease evaluation because the deer are so twitchy in experimental rooms, right? It's hard to count respiratory rate because they're hard to look at. And in the wild, hunters aren't coming in saying, yeah, this deer was sick for sure. But humans also don't spend a ton of time staring at wild deer. There's 30 million of them in the US, and we're really only seeing such a small snapshot of that. And so it might just be an accessibility thing. I'm not sure.
Yeah, there is also, because humans, we express, like humans have symptoms, but animals, we're not sure if the deer are having any, like, we're not sure if deer have symptoms because they're not expressing that.
Right.
And we also have to remember that the deer are prey. They are animals that are hunted by, naturally, they were hunted by wolves, by cougars, by bobcats, small deer. So they are prey animals. They cannot show signs of disease. It's not natural. It's part of their evolution. So like Dylan said, it may also be a problem of sample size.
So I guess what's next? So Leonardo, what are the next questions that you are addressing in this ongoing, I guess, zoonotic infection now with SARS-CoV-2 in deer?
Yeah, there's the thing that I'm most curious about right now is to see what will be the outcomes of this current hunting season. We want to see if these variants that we found are still present, and if they have spread to other counties, if some of these variants became more established than others, alpha, gamma, or delta, and also if the initial lineages, if the B1-like lineages also spread, because we found very few sequences in the last hunting season. But then, yeah, this is the thing that I'm most curious about right now, to see how these variants are going to spread, not only in New York State, but in the other states where SARS-CoV-2 was detected in deer.
Right. I mean, because at least in mouse models, the Omicron variants didn't do so well. So they were much more attenuated than potentially in humans. And so you kind of wonder, maybe they're not as fit, as it were. They won't spread as much in deer themselves. Dylan, what about you? What's next?
Yeah, well, first of all, you raise a great point about upcoming variants, right? The fitness landscape in a human population that's largely vaccinated is so different than what we might see in a wild deer population. So it's a great point in that as we move into establishment in both humans and deer, that fitness landscape is really going to start to diverge, I think. And yeah, like Leonardo said, with passing time, as we get to that establishment, I think there's so many great questions, right? When we started looking at our sequences from this past season, right, it's like, do we have the ones we found in the first detection? Like, do we have more viruses from these clades, right? We didn't, but that was the big question. And it's sort of points to this still sporadic nature of the spillovers. Looking at which of these are really going to hang on is a huge question. And I think time is going to have to be the true tell for that. On top of that, I think talking about, obviously everyone wants to know how they're getting it. That's a huge question. And also seasonality dynamics, right? I think largely, even though wildlife are hard to sample, white-tailed deer are truly one of the most accessible wildlife species because there's a lot of population control programs. And quite frankly, people love hunting them, right? And so a lot of these samples, I would argue maybe even all of these samples so far are really dependent on the hunting season, because that's when these cull programs and hunters are going out and taking deer.
Mhm.
It also corresponds with post-peak mating season for deer, right? Because they go into this rut and they're more active. And so that's when they're easier to hunt. But if they have this potentially drastically different contact structure associated with their mating season and then post, is that inflating the maintenance prevalence that we're detecting after that, right? So what does prevalence look like in white-tailed deer in June? I certainly don't know, and it's a very difficult question to answer, but year-round surveillance is something that is important in all wildlife reservoir species. It's unfortunately a really difficult gap to bridge. It highlights the challenges of surveillance, and surveillance also happens to be really difficult to get funded in a modern funding landscape, right? A lot of funding agencies, there's been this move to hypothesis driven science and a lot of basic science, which is awesome. And it answers really great questions, but surveillance from a public health perspective is really important. And unfortunately, in a lot of animal species, it can get pretty expensive, and it can be difficult to get funded if you don't have a very specific research aim. So I think the establishment of this field and trying to bridge all those gaps and answer those questions is going to be important in the coming years.
Right, right. And Leonardo, so what's next for you scientifically? What is I guess the next step in your scientific path?
Yeah, so now right now I'm a research associate at the Animal Health Diagnostic Center in Cornell University. So I'll be naturally working with wild animal samples whenever they come to the laboratory. That I'm really interested about metagenomic sequencing. I think this is one of the best tools to detect viruses, untargeted detection of viruses. So I really want to get deeper into that field, into a broader virus detection in wild animals.
And how do you, so I guess we were talking on the podcast last time with people that were doing sort of air and wastewater surveillance. How do you do metagenomic sequencing in sort of wild animals? How do you, what's the sort of methodology, or how do you get those samples?
Yeah, the easiest way of getting samples is when animals die naturally or they're hunted. But then there's something is not really common to be used is non-invasive sampling. And I think this is something that must be, this is one of the ways to get samples from wild animals. Then we have the bottleneck of sample quality, because samples would be exposed to the natural conditions. But it's very, like Dylan said, it's very challenging to obtain samples from wild animals.
And Dylan, so what's next for you?
So I'm actually a fellow in the NSTP program, which is the National Bio-Agro Defense Facility. It's the brand spanking new USDA facility they're building in Manhattan, Kansas. And so they've created this scientific training program to recruit terminal graduate students to fill expertise gaps when they open that new facility. And so I most of my, at least the last couple of years, my graduate education has been supported by that fellowship, which is really awesome, from USDA APHIS. And so that'll see me out to the Foreign Animal Disease Diagnostic Lab for full-time work once I finish. So I'll be working a little bit more on the animal side compared to the public health side right now. But because it's a BSL 4 level facility, they've added a lot of zoonoses to their the APHIS mission statement and things like that. And so I look forward to still working from a One Health perspective, but taking a look at transboundary and emerging animal diseases. So I'll be headed to the government sector, a little bit of public service in my future, but I look forward to being able to do a lot of applied problem solving. I think that's one of the real strengths of working in government. I love research, but I think, using the scientific process to do more applied problem solving, I think sounds really cool. And so it's not something I have a lot of experience with right now, but I look forward to heading out there.
Right. And then I guess just to finish up, I'd like to ask people sort of how they first became interested in science and virology. Is there a particular teacher or moment or something that kind of started you off on the path that you're on now? Leonardo, why don't you take that?
Yeah, for me, I think one thing leads to the other. So since I was born, since as I remember as a human being, I was interested. I always liked animals, especially wild animals. So from wild animals, I got into veterinary, and I always liked birds in special. Nowadays I'm still consider myself a bird watcher. So from poultry, I got into vaccine viruses, and then from the vaccine, I really found interesting the vaccination in poultry industry. So from that, I got into virology, and then from that, in virology, I got really interested about next generation sequencing and metagenomic analysis.
Cool, great. And Dylan, how about you? How did you first sort of start going down this path?
First of all, I love that Leonardo is a bird watcher because I'm a birder myself. I'm a huge fan. I just got a magnet on my fridge that says bird nerd on it. So I also love birds. Much like Leonardo, just like really into animals as a kid. I always loved the zoo. Was a fan of all of our pets in the house growing up, and just a very curious child. I'm an answer seeker. So growing up, I don't have academic parents, and so it took me a little bit to sort of navigate the waters of graduate school. You hear scientists as a word, or like researchers, in the public, but it's not super clear for somebody not in academia what that means, or all of the different ways you can actually do science. And so it took me a little bit of time to navigate that. I would say, shout out, you said a teacher. I'm going to shout out my AP bio teacher from my high school, Miss Clymer-Smith. So I'm from New Jersey originally, and I always liked animals. I always knew that I liked science. And so I took a lot of the more advanced bio classes, Mr. Hausman, anatomy and physiology at my high school. And she really did an awesome job, Miss Clymer-Smith, of like kind of letting us foster scientific curiosity in a more free way. Like as a senior in high school, we had a couple of projects that were true, like ask a scientific question, like how do you want to answer it? And as a 17-year-old kid, like that was really cool for me. And I think it really sparked something for me that led me down a path where then selecting universities I wanted to go to, like having accessible undergraduate research opportunities was like top of my list. It was something I definitely wanted to do when I got to college. And so when I got there, I got engaged in undergraduate research really quickly and just really found that I loved it. I loved it a lot more than some of the like clinical medicine experiences I had before. And so it was a super clear arrow pointing me towards this sort of research path. I had plenty of great professors in college. Dr. Aaron Lindstedt did a really good job of shepherding me along my way and helping me figure out how to apply to grad school and find an advisor and things like that. So it's just honestly just been super fun.
Well, thanks so much for talking to us about white-tailed deer and SARS-CoV-2, and a little bit about yourself as well.
Yeah, thank you so much for having us.
Yeah, thank you.
This has been Let's Meet the Virologist, a podcast about people who study viruses. This is your host, Larissa Thackeray, and thanks for listening. You can find us on Google, Apple, Amazon Music, and other podcast providers, or at lmtv.podbean.com.